Okay, so my name is Dr. Lauren Seyler. I am a second-year assistant professor in the biology program here at Stockton. And today I want to talk to you about some of the work that we have been doing in my lab here at Stockton and our future plans using a new type of DNA sequencing technology. So first of all, I want to come right out and say it. I'm a microbiologist. And when I tell people that I'm a microbiologist, they either have no idea what that means or they imagine images like these. These are the kinds of pictures that come up if you Google microbiologist. And it's lots of people looking down microscopes, looking at petri dishes with microbes growing on them, and wearing masks and things of that nature. So in order to define what I do and what we do in our lab, it's helpful to take a step back and talk about what microbes are. So a microbe is life that's too small to be seen with the naked eye. So this includes things like bacteria, another group of organisms called archaea, which are very similar to bacteria physically, but genetically very different, viruses, many types of fungi, and also protists, which are single-celled like bacteria, but much more complex, things like amoebas and protozoans. To most people, when they hear the term microbes, they immediately think germs and they think disease. And this absolutely makes sense because over 17 million people die every year from infectious disease, according to the World Health Organization. So when people think microbes, they think disease, they think infectious disease. And we started studying microbes in order to better understand infectious diseases and how to treat them or prevent them. So most of what we know about microbes comes from our study of infectious disease. But germs really aren't the entire story of the microbiological world. There are about 1,400 known species of human pathogens, but compared to the total number of microbial species worldwide, it's not a completely agreed upon number. A low estimate would be about 120,000 different microbial species, but some estimates put it in the trillions. There are roughly 5 million trillion bacteria on Earth total. So to put that in perspective, that's 1 million times greater than the number of stars in the universe. So we really live in a world ruled by microbes, and we're surrounded by this invisible world. And the vast majority of these organisms are not causing disease, and in fact, can be very beneficial. So what are they all doing? Well, lots of different things. They can be producing oxygen. In fact, microscopic algae in the world's oceans produce about 50% of the total oxygen in our atmosphere. That's a lot more than the Amazon rainforest. It usually gets all the credit. They're providing important nutrients to plants, including things like nitrogen. They decompose dead material, which is very important for taking all of the nutrients and decaying biomass, dead things, and making it available to other living things. They remove pollutants, and a lot of research is being done on how to use microbes in order to bioremediate places that have been polluted, industrial sites or sites that used to be mined. They digest your food. A lot of your digestion is carried out by the microbes that live in your intestines. And they also defend your body from infection. Your body is colonized by millions and millions of bacteria. For every cell in your body, there are 1 to 10 bacterial cells. The number is not exactly agreed upon, but it's anywhere between 1 and 10 bacterial cells for every cell that is human. And these microbes are there defending your body from foreign invaders that could cause dangerous infections. So how do we study life that's too small to see? I mentioned microscopes before, but if this high estimate of the number of microbial species is accurate, then that means that we haven't discovered 99.999% of all of the species that are out there. And this is because most of them can't be grown and isolated in the lab. Looking down a microscope doesn't really help us much because, just to put that into perspective, Say you were looking at a gram of dental plaque under a microscope. It contains 100 billion bacteria, which is equivalent to every human being that's ever lived. Imagine trying to look at that under a microscope and make some sort of sense out of it, particularly when a lot of microbes look very similar under a microscope, but may have completely different properties. So when it comes to my research, I don't really use a microscope very often, if at all, because it just doesn't really tell me much about all of the complexity that exists in the average microbial community in the environment. So these are some pictures from my graduate and postdoctoral work. I've studied microbes in the open ocean, in the deserts of Oman and the Samal ophiolite, and in California, and just about every type of ecosystem you can imagine, from the very wet to the very dry, and the very extreme as well. But I don't really use a microscope to study microorganisms. What I do primarily is look at DNA. DNA is the instructions on how to build a living thing. And if we begin with those instructions, we can start to get at not only the identity of all of these microbes, but what are their capabilities? What are they able to do in the environment? And what services are they providing? So just a brief overview of how the language of DNA works. DNA is a language made up of 4 letters. These are bases, A, C, T, and G. Every word is made up of 3 letters, and these words provide the chemical instructions for making proteins, which are the machinery of the cell that perform all of the functions of a living thing. The example gene that I have here is the gene for green fluorescent protein from jellyfish. And so this is a very simple gene, and you can see that it's just repeats of all of these different letters that give the instructions for making this protein that makes jellyfish glow. So what we do is we take these DNA sequences that we acquire in the laboratory and compare them to databases of known genes to start annotating them and make sense of the genetic capabilities of the organisms that we're trying to study in the environment. So really, we're just extracting DNA wholesale from these microbial communities, sequencing it, and then comparing it to these databases to try to figure out what genes are present and identify microbial species. So there are a couple of different ways that we can take DNA and sequence it. And the 2 methods that were available at Stockton when I came here were Sanger sequencing, which is the original DNA sequencing method that was invented in the late 70s. This works by making a bunch of copies of the DNA molecule with random stops, which creates a bunch of fragments of DNA that can then be separated and interpreted. This method is really best for studying individual genes and at very small sample numbers. It doesn't scale up very well. Next generation sequencing was invented in the 90s. And we have a next generation sequencer here at Stockton that uses what's called ion torrent technology. Next generation sequences are generated by reading the DNA as the copy is being made. These can be scaled up quite a lot. They generate much shorter sequences of DNA than Sanger sequencing does, but you can produce a lot of it. So you end up producing huge amounts of data in short amounts of time. Very, very large data sets that can then be interpreted by computer programs. So if you're looking at the DNA of an entire microbial community, the standard method is to take that DNA, break it up into a bunch of little pieces, sequence all of the pieces, and then try to figure out where those pieces overlap to assemble genomes, whole collections of genes. To give you an idea of how complicated this can be, I have an analogy. Let's say you've never seen the show Game of Thrones, and someone gave you several thousand copies of the show in a digital format, but each copy had randomly been cut into 10-second clips. And you had to figure out what the plot of the show was from these 10-second clips by watching them all and figuring out where they overlapped and what order they were supposed to go into. Frankly, it sounds like an absolute nightmare. And that's one show. So imagine that one show represents one species. Now, if you were trying to characterize a more complex community, imagine trying to watch a whole bunch of shows in this way at once and figuring out which show is separate from another show. So if you notice a certain groups of actors are all in a certain number of clips, you would pool all of those clips together and that would represent one genome. And then another group of actors or different plot or different costumes or whatever clues you would use would help you figure out how to separate these shows and then line up their plots in order. Obviously, we don't do this by hand. We have computer algorithms that do it for us. But even when using a computer, this can be a very, very difficult prospect. So a new type of technology, which is sometimes called third generation sequencing, became available in the last 10 years. And I recently acquired a sequencer that uses this technology. And much like me at Stockton, it's sort of the new kid on the block when it comes to DNA sequencing. So it works by passing the DNA through pores, which are embedded in a synthetic membrane. So these pores are billionths of a meter across, and the DNA molecule is passed through this pore in the synthetic membrane, which has an electrical current applied to it. As the DNA passes through the pore, each base, each letter in the code causes a change in the electrical field. This change is interpreted to provide you with a DNA sequence. The really incredible thing about this technology is that it provides you with much longer reads. So to go back to our Game of Thrones analogy, it's like watching 7-minute long clips as opposed to 10 seconds, which makes interpreting your data significantly easier. But also, you don't have to copy the DNA in order to sequence it. You can read it directly. So this greatly reduces bias in sequencing and possibilities for error in copying. The other truly amazing thing about this sequencer, and I brought it with me if anybody wants to see it, it's incredibly portable. So the unit that I have is about the size of a small book. There are ones that are essentially a thumb drive that you can plug directly into a laptop. And so these sequencers can be taken around the world to do sequencing directly in the field. So rather than taking a sample and having to freeze it and transport it back to the lab, you can sequence it right there. And this is incredibly useful. People have used this technology to perform sequencing in West Africa, in East Africa, in Tanzania, Uganda, Kenya, in the Canadian high Arctic, in Antarctic dry valleys, and even on the International Space Station. So the capability to perform DNA sequencing right when you need it, right where you need it, is an incredible technology. And I imagine that very soon it's going to be spread even further into places like your doctor's office, where they can sequence your genome right there while you're waiting. So where will we go, the Seyler lab, the deep lab here at Stockton, with this technology? Well, we've already started a project looking at bog iron in the Pine Barrens. So bog iron is this very dirty iron ore that provided a thriving industry in the Pine Barrens in the 18th and early 19th centuries. And it's formed when iron-rich acidic groundwater in the Pine Barrens that's full of dissolved iron comes up to the surface and comes in contact with oxygen. And the oxygen in the air essentially turns that dissolved iron into rust. Bacteria are part of this process. They actually accelerate the process of oxidizing the iron. And there are other microbes that live on this oxidized iron that use it in their respiration. So they're essentially breathing oxidized iron. So there are microbes pumping iron right here in the Pine Barrens at these historic sites. So we started doing a little bit of sequencing and looking for different types of microbes in these environments. And we started with some samples right around Lake Fred here on Stockton campus. And what we found is that almost 50% of the microbial community associated with these iron oxides was cycling iron in some way, either directly oxidizing it or reducing it and using it in their respiration. In addition to that, we have a couple of other projects in the pipeline. So I have some samples from when I was a grad student. The picture of me earlier on the ship is actually from when I took those samples, where we may be looking at microbial relationships in the deep Caribbean and how microbes are involved in the breakdown of carbon and the storage of carbon long-term in the deep sea. We're also looking at a carbon storage project in salt marsh sediment. And I have an undergraduate student who's interested in looking at fungal diversity in white cedar swamps. She approached me with this project idea, and she wants to look at how changes in the vegetative regimes in the white cedar swamps affect the diversity of the fungi that live around the roots of these trees. So we've got a lot coming up, and I'm very excited to share it with you in the future as more develops and we get more data. So I just want to acknowledge all of the students who have been working with me since I started my lab last summer. I have an amazing group of students. One of them is here, Kayla, who has been doing some awesome work on this bog iron project with us. And there's more to come for sure. Some enrichment culturing, I hope, and some other sequence projects we're working on. So we should have some data soon to show you. But that about wraps up what we have for today. So thanks everyone who came, and I'm happy to take questions.
I have a question.
Yeah.